Okay, so welcome back to this next video in which we are discussing the LDL receptor. Okay, so we've discussed that when LDL binds to the LDL receptor, what follows is a process called receptor-mediated endocytosis. Okay, so the LDL receptor, along with its cargo, binds to the adapter protein, which is the LDL receptor adapter protein 1, and that then recruits clathrin. Clathrin polymerizes to form a buckyball, which then pinches off the membrane, and you produce a clathrin-coated endocytic vesicle within the cytoplasm of your cell. What then follows is the uncoating, where you lose the clathrin coat, and you then have an uncoated endocytic vesicle, which will then move to an intracellular organelle known as the early endosome, and it will fuse with the early endosome to put the LDL receptor with its cargo, the LDL molecule, into the early endosome. It doesn't remain in the early endosome for very long. It's then trafficked to the late endosome again by another vesicle, which is pinched off the early endosome membrane. Okay, in the late endosome, what then happens is you have a very acidic pH within the lumen of the early endosome, which causes the LDL receptor and the LDL to break apart. And what can then happen is that the LDL receptor is then recycled to the plasma membranes. You pinch off vesicles from the late endosome and you rescue the LDL receptor, take it back via another vesicle to the plasma membrane. Meanwhile, the LDL molecules, they also get pinched off into another vesicle, which then goes to the lysosome, which contains all sorts of enzymes which will break down the things that are within this LDL molecule. So, the apolipoprotein B100 will be broken down by proteases within the lysosome. The phospholipids, which are mainly lecithin molecules, they will be broken down by enzymes within the lysosome to yield long-chain carboxylic acids. Okay, you will then have the cholesterol. That will be spared, okay? So the cholesterol doesn't have anything happen to it. But the cholesterol esters, they do get broken down into cholesterol and free fatty acids. Okay, so the free fatty acids that you get, those will just be used for beta oxidation or other metabolic processes. But the main thing that we are delivering to the cell is cholesterol. Okay, so let's now discuss which sites within the body actually have LDL receptors on them. Okay, so we'll start with extrahepatic sites. So one of the key sites which actually has LDL receptors on it are liver cells, but we'll come back to those and discuss them at the end. Okay, so we'll start with extrahepatic sites. Okay, so firstly, you have adipocytes and muscle cells. So adipocytes and muscle cells both have LDL receptors on their surface, and therefore will endocytose LDL molecules, and therefore will be getting these deliveries of cholesterol into their cytoplasm. Okay, now what do they use the cholesterol for? Well, basically, cholesterol is an essential part of the cell membrane. So these two cell types are going to be using the cholesterol to make plasma membrane. Now, a slightly more interesting example. Okay, another extrahepatic site which has got an LDL receptor on its surface are the cells of the adrenal cortex. Okay, so basically, let's start off with a little picture of an adrenal gland. Okay, so basically you have two adrenal glands, one atop each of your kidneys. Okay, and the adrenal gland can be split into two major portions. So there is the adrenal medulla, which is responsible for producing adrenaline. Okay, and then there is the adrenal cortex, which produces a huge number of steroid hormones. So this is the adrenal cortex. Now, the adrenal cortex can be split further into 3 separate layers. Okay, so let's split the adrenal cortex. Let's say this is the adrenal cortex. It can be split into 3 layers. The outermost layer is known as the zona glomerulosa. Okay, so this is the zona for zone, and then glomerulosa. Okay, and basically this is responsible for the production of the mineralocorticoid aldosterone. Okay, so this produces aldosterone, which plays a crucial portion in the renin-angiotensin-aldosterone system. Okay, then the middle layer of the adrenal cortex, this is called the Zona Fasciculata. Okay, so this is the Zona Fasciculata. And basically the Zona Fasciculata creates glucocorticoids, the major example being cortisol, okay, or hydrocortisone as it's also called. So here is the Zona Fasciculata, and this creates the glucocorticoid cortisol. Okay, and then the final layer down here, this is known as the Zona Reticularis. Okay, and the Zona Reticularis is responsible for the synthesis of sex hormones. Okay, so sex steroids, androgens, and the like. Okay, so this is in red here. Right, so all of these hormones that they're secreting, aldosterone, cortisol, and sex hormones, these are all examples of steroids. So they have that steroid structure within their structures. Okay, so these 4 carbon rings joined together, 3 6-membered carbon rings and 1 5-membered carbon ring here. Okay, so where do they get this core steroid structure from? Well, basically the starting point for all of these layers synthesis of steroid hormones is cholesterol. So basically all of these cells have on their surface LDL receptors. So the cells in all 3 layers will have LDL receptors on their surface so that they are getting deliveries of cholesterol which they can then use to manufacture the steroid hormones. Okay, right, so that's a key extrahepatic site where you have LDL receptors. So let's now discuss the liver, okay? So, remember the liver cells, the hepatocytes, they were originally producing these VLDL molecules. So they originally put into the VLDL all of the cholesterol and all of the cholesterol esters that are now left over in these LDL molecules. Okay, so hepatocytes do indeed have LDL receptors on their surface and will reuptake the cholesterol that they originally put into the VLDL molecules. Okay, so hepatocytes are continuously removing LDL molecules from the blood. So they're getting the cholesterol back again. What do they then use the cholesterol for? Well, cholesterol is a key starting point in the synthesis of the bile acids. So all of the bile acids are made from cholesterol derivatives. So they will use cholesterol to make bile acids. And if they've got too much cholesterol, what they can do is they can actually store it. So they store it as cholesterol esters. So this is a little bit sort of redundant, since they originally took the cholesterol esters in from the LDL molecule, broke them back down into cholesterol and free fatty acids, and now what they're going to do is resynthesize them back into cholesterol esters, but it's the way it's done. Okay? So what happens is they use an enzyme called ACAT, which stands for acyl-CoA cholesterol acyl transferase. Okay, so the first A is for acyl coenzyme A. The C is then for cholesterol, so the C is not for CoA, the A is for acyl-CoA. The C is then for cholesterol, the A is for acyl, and then the T is for transferase. And basically this adds an acyl group back onto the cholesterol molecule to create a cholesterol ester. And then these cholesterol esters can then be stored in cholesterol droplets within the hepatocyte. Okay, so hepatocytes can store cholesterol within cholesterol droplets, basically. Okay, right. So that now concludes our discussion of the LDL receptor. What we now want to look at is when all of this goes wrong. So we've already seen this example of autosomal recessive hypercholesterolemia, which results because of double hit of the genes for that LDL receptor adapter protein 1, which completely stops the endocytosis of LDL molecules out of the blood and results in far too high LDL levels within the blood. Okay.